All right. Hello, everyone. We will go ahead and get started. Welcome to the Soil Health Digital Cafe. This is hosted by the Soil Health Nexus. For those of you who don't know us, we are a university-led team dedicated to increasing access to research-based soil health knowledge, extension, and resources. We have representatives from all 12 of the North Central region states, and we have a number of initiatives we've been working on focused on extension and education, and many of those can be found on our website, soilhealthnexus.org. We have a soil health toolbox on there full of soil health resources, including information on soil health's chemical, physical, and biological properties. So that's a great resource for you. And this digital cafe series, if you haven't joined us before, we host it monthly and feature a different speaker talking about soil health. How it generally works is we're going to have the presentation followed by Q&A. So please put your questions in the chat box, and we'll have Q&A. And then after the Q&A, as time allows, we will break out into some breakout rooms for some small group discussion. And we have those breakout rooms formatted kind of based on your role. So researcher, educator, agency, farmer, student. So you can choose the one that makes the most sense for you and have some discussion on your reactions to today's presentation based on based on where you're at, and some also discussion if you have any other topics you'd like us to include in future digital cafes. So I'm very excited for today's presentation. It's gonna talk about activating the soils and starting at the surface with a focus on measuring soil and soil's biological activity and measuring soil life with Alan Franzluebbers with the Agricultural Research Service. So we're very excited to have him today. I am going to go ahead also, one thing I forgot is I'm going to go ahead and launch our poll here today. That is a little bit of information on you. All that information is anonymous, but we ask it as a federal service as a part of the extension in order to ensure we continue offering these educational properties. So I'm going to go ahead and launch that poll and then hand it over to Alan for his presentation. Alan, thanks so much for joining us today.
Thank you, Anne.
All right, go ahead and share your screen, Alan, whenever you're ready.
Okay, very good. Thank you, Anne. Well, it's a pleasure to be here. As indicated already, I'm Alan Franzluebbers. I grew up on the land in Nebraska, so I'm familiar with a little bit of the North Central region. I'm already feeling cooler today just by presenting to the North Central region. Thanks for the cooling effect that occurs. Well, conservation management brings soil to life. Basically, this is an important concept, is that we need conservation management. It's important for us because it's the carbon in soil that really is the food source for those organisms. So measuring change in soil life needs to be simple and robust, because otherwise we're not really going to make much progress. We can make a little bit of progress with complex methodology, but really we need some simple methodology too. And that can really start with how much carbon is in the soil and what it does for the organisms in that soil, as well. And those organisms could be the microorganisms, or they can be the plant roots, and also the macrofauna that live there too. So what is soil health? Well, the key feature, it doesn't matter whether your soil is red or black. It doesn't matter if it's yellow or if it's got a gray tone to it. It's really about how the roots proliferate and penetrate the soil and access nutrients, water, all the things that really soil is supposed to function for us. And so getting good rooting activity is important. So simple definition: continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. And really it's about those roots, those plants, the plants really taking up CO2 from the atmosphere, using the energy from the sun. That's the starting point. So there are a number of important functions of soil, and I think that's important for us to keep in mind. We want our soils to be able to produce plants and food. It's important. We as people on the land are also a part of the ecosystem. So we have to make sure that food and fiber production does occur. And we should understand that we should be within balance within our ecosystem boundaries. But we also want to cycle nutrients and protect water quality. Soil is important for doing that. The soil should be able to store carbon and nitrogen and enable an animal habitat, whether that's for those that can crawl around or run around on the soil, or are just simply basically stationary within the soil. And so that animal habitat is important. And then we want it to do all sorts of other things, like supplying that water and the plant growth promoting compounds, serving as this reservoir of biodiversity for these various organisms that are in soil, and also to able to avoid toxic accumulation of elements. And that could occur in some situations. And so the organisms in soils are doing that. And so the soil is really important for all of these many functions. But there are a few key indicators. I think we've made it a little bit complex sometimes. I mean, it's good to be complex. We want to understand the complexity, but we also should have this overarching theme of simplicity. And somehow we need to strike a balance. We need some people to be into the diverse, complex nature of soil, and we need other people to be more on the simple part. And so maybe I'm going to share with you a little bit about a simpler approach. Hopefully that it'll provide some in-depth answers, but it's a simpler approach. So we have had for several decades the inorganic nutrients being measured in our soil testing labs. So state soil testing labs have provided that service. Commercial labs do that, and we should continue to have those chemical nutrients being determined. We can see the physical attributes of soil, and unfortunately we don't have a whole lot of soil testing for that. It's pretty obvious for us that aggregation is important, but we'd really want to know about how water infiltrates soil, whether the water is infiltrating properly or if it's running off. And that's a big thing, but that takes oftentimes field scale measurements, and so it's a little bit more complicated, but it's an obvious thing. We do know that soil organic matter is this key biogeochemical feature that is under all soil health analyses, that soil organic carbon or nitrogen are important key elements that drive many of the ecosystem processes. So that's an important part. And then the biological component, of course, we could get into the actual physical, the microbial community structure or the biomass of the microbes, but we can also determine this from a simple test of soil test biological activity. And so the activity of the soil tells us about what are the resources there, are the conditions right for this activity, and then that lead to many other functions of soil, such as the biodiversity, as well as nutrient cycling and storage of carbon and nitrogen. So what is soil test biological activity? The way I've defined it, and the way I've developed it more than 20 years ago already, a measure of the organic resources in soil that are readily digestible by the community of soil microorganisms at the time of sampling. So this is an important definition, because whenever we take a sample from the soil, of course, we've now extracted it from its natural environment. And so there's always some handling features about it. And the key issue was that we knew that the soil had to be dried, because that was the routine soil testing, that paradigm that is out there. And that if we could do that, then that would be useful for us. So we actually do dry the soil and re-wet the soil. And this was going against the nature of soil microbiology, but And in fact, we did it anyway, and found that this flush of activity in the first few days was very important and highly related to the activity that occurs later in incubation. So we need to determine that CO2, and we do that simply by this diagram. First, I'm going to draw your attention to the diagram, the picture on the left, in basically a canning jar with some soil in it and an alkali trap. It's sealed, and then the soil respires once we add the water. And then basically we get the results on the right, where we have wetted that soil. We create this flush of activity that occurs for the first few days, and it may depend on the soil type, how long that may occur. But in general, the first 3 days has been relatively good for capturing most of that flush of activity. And then over time, such as after approximately 10 days, we get a more of a linear release, and that we can relate to the basal soil respiration. And in this diagram on the right, we see that the soil depth does matter. And so we want to make sure that we analyze the soil depth that we're really targeting. And the depth distribution there shows that the resources are really at the surface. And so characterizing that surface is really important. So the flush of activity during the first 3 days, here circled in these 3 points, indicates a lot to what was happening later on. But there are some key considerations, and these are just generally, protocol determined. And if you're not into the methodology, I'm just going to go through these quickly. But essentially, we definitely have to get a representative sample of the field of influence. So we want to make sure that we get enough cores in the field, or a portion of the field that we're interested in, so that we can actually get a good estimate. We want to have a defined depth, because as you see on the diagram on the right, there are the depth really does matter as to the quantity that is released. And then we protocol wise, we oven dry the sample at 55 degrees Celsius. And of course, this is considered an elevated temperature, but it's also a realistic temperature. And some soils in nature are exposed to this high temperature. Certainly not all soils, but at the very surface, when the soil is exposed to the sun, it can easily reach that point or even higher. And we want to sieve the soil coarsely, partly because I've always been interested in aggregation as well, but also because when we incubate the soil, having these larger voids in the sample allows the water to penetrate well, and it redistributes itself really nicely just because of capillary action. So we want to re-wet to 50% water-filled pore space, and that is determined by currently we're doing it by having a standard scoop size, and then determining the mass, and then from that we can actually calculate how much water to add. And we incubate at a 25 degrees Celsius or 77 degrees Fahrenheit for 3 days, and then we try to determine the amount of CO2 through an alkali trap. We put in some barium chloride to separate that out, and then we titrate the remaining acid, or the remaining alkali, with an acid. And that's the way that's determined there. So I've had a lot of collaborations, and I think it's important for any research group to kind of expose others to what are the collaborations. And so extension has been a key component for me, but also some of the actual agricultural groups, such as the Soil and Water Conservation. The Cotton Incorporated, we've worked with them considerably over the years, and recently more the Forage and Grassland Council of North Carolina, and water quality people are very much in line, and NRCS is a key partner throughout all of this as well. But the real partners are the farmers themselves. And these farmers have been really gracious for my research part, and they are allowing me to come on their farm. They want to know information, so they're willing to do a few things. Of course, I'd like to be, I would say, or invisible, let's say, that I don't want to imply or take too much of their time. I don't want to also do too much that is different from what they do. I'm trying to take their farm situation and learn from what I can from that. So I want to give you a little flavor of some of the conservation effect on soil biological activity. And in this case here, these are 3 depths, 0 to 10 centimeters, 10 to 20, and 20 to 30 centimeter. and showing that under conventional tillage with no cover, there's a higher activity near the surface and it declines with depth, and that's what we would expect. When we have no-till and no cover, we elevate the amount of biological activity at the surface because we're not disturbing the soil, and then those carbon resources are essentially preserved a little bit more at the surface, and you can see that the biological activity also decreases with depth, and but it tends to be a little bit higher than under conventional till with no cover. And then finally, when we have no tillage with a cover crop, either single species or multi-species, we can elevate the activity near the surface even more. And sometimes it increases it with depth, but it doesn't always. And partly this is in the southeastern U.S. We have a very warm and mild climate, so it's warm and wet. And so we have decomposition proceeding most of the year. These observations kind of led me to the idea that we need to make sure that we are covering our fundamental bases. So the idea was that with soil test biological activity, we're building up resources, and it should really, and we then measured the nitrogen mineralization in the laboratory, but we wanted to really find out, does this really lead to differences in plant growth? And then I'm going to lead you through this. We did a series of greenhouse trials, and then we also went to the field. So that's where I'm going to lead you towards. But basically, we would take samples from different farms, different research stations across North Carolina and surrounding states, and we'd put the soil into these tubes and only give it water and 5 seeds of sorghum Sudan grass, and allowed the sorghum Sudan grass to grow for 5 weeks and see what kind of growth would occur. If the soil had enough nutrients from the mineralization of organic matter, then the plants would grow well. And if they didn't have anything there of organic matter that was biologically active, then there just wouldn't be much growth. And so these are some of the soils where they were black, they were yellow, they were a little bit grayish. And so they were also, in this case here, this would be a sandy soil with very few aggregates. And then we had those that had more aggregates. And we would test surface soils as well as subsurface soils. And these are the seedlings emerging, and we covered the surface with some regular quartz sand just to avoid contamination from any splashing, although we did water from the bottom so that we would avoid that as well. And this diagram here shows that plant dry matter production was a strong function of net nitrogen mineralization. So we took each of those soils, we would determine its net nitrogen mineralization. This happens to be in the coastal plain region where we have coarse textured soils. And we see that nitrogen mineralization was the key factor in really determining plant dry matter production. So nitrogen was the limiting factor, and nitrogen mineralization is a biological process. So it's something that we had to have that in the soil. So with this, we can actually determine that the nitrogen mineralization leads to a certain amount of plant dry matter production. And so we have this linear association. We can actually start quantifying what is the relationship between a potential plant dry matter production and nitrogen mineralization. We add in some soils from the Piedmont region. This is a little bit hillier landscape. It's a higher elevation. It's farther away from the coast. And we see that there's also an association between plant dry matter production and nitrogen mineralization. It's not exactly the same as in coastal plain, but it has its own association. And adding in the Appalachian region, the Blue Ridge Mountains, and we see another association. But essentially, if we put them all together, there was no difference. We had plant dry matter production that was highly related to net nitrogen mineralization. So that net nitrogen mineralization is an important aspect. And then again, we can actually make some quantitative determinations. This was in the greenhouse. We really didn't need to make that quantitative determination because we're not really growing any plants in the greenhouse in this case. We wanted to take it to the field. But so in this case here, I've replaced these associations with plant available nitrogen, which is the sum of residual inorganic nitrogen plus the mineralizable nitrogen, and related that to soil test biological activity. And you can see that there's a very strong association there as well. So the reason that we have plant available nitrogen, at least the mineralizable portion, is because of the soil test biological activity. That's driving the mineralization that occurs there. And so if we replace the x-axis with soil test biological activity and associate that with plant nitrogen uptake in the greenhouse, very strong association there as well. And so we're pretty confident that the reason we can have plant nitrogen uptake or an enhanced nitrogen uptake is because of the biological activity itself. And we can look at that in different soils from, now these are taken from different soils where we have biological activity associated with net nitrogen mineralization and very strong association there too. Now there are some deviations, and certainly net nitrogen mineralization is not a one-way process. There's also immobilization that can occur. And so there are some factors that could make the association weaken. But in general, we find this high association, and in fact, just to point out some of the details here, this 27% of the soil test biological activity, this is very strong. It's occurred in more than a half dozen studies where different soils, different conditions, that's what we're observing. And oftentimes we get 70% or up to 85% association between these two parameters. So feeling pretty comfortable that this is a stable relationship. When we took it to the field, these are some of the results that occurred. We take this soil test biological activity, which is very simple soil measurement, and then related it to the in-field grain yield response to the initial dose of nitrogen fertilizer. This is, if you can imagine, and I'm going to try to explain it a little bit more detail, but it's the yield response if you didn't apply any nitrogen to, say, a corn crop, and the first yield response that you could get from any dose of nitrogen, this would be the instantaneous rate that would occur. And we would expect that there would be a high yield response with very low biological activity, because as I've shown you in the greenhouse, we get very low plant dry matter production with that. And so we need nitrogen fertilizer in that case. So this is an amount of nitrogen basically that we would be applying. Whereas if we had higher biological activity, we wouldn't need as much nitrogen. And so this was an important finding, really to bring us to from a laboratory determination only to what actually could be occurring in the field. And this curve is really related to the yield responses. We might say that this is a classical curve here, where we apply a unit of nitrogen, say 40 or 50 pounds of nitrogen, and we get a very large yield response. And then in another situation, we might have a field where there's actually a pretty good amount of biological activity there already. We apply some fertilizer and we get a modest amount of yield response to that application. And then in the final case here, where we would be at the very right part, far right part of this curve here, we'd have a yield response that might look like this. And all three of these yield responses to nitrogen fertilizer application are occurring. When we do field trials, we see many times where we get very little yield response, or we get a very large yield response. And so this is trying to figure out why is that occurring, and that we can isolate it, at least for the most part, in many of our field trials in North Carolina and surrounding region, to this biological activity. And that gives us good information where maybe we could use it as a predictive value. And this is explained in a recent paper that was published with my colleagues here, where basically wanted to share with them with others the idea behind this, because this occurred over several years. This was a kind of a 10-year project that occurred. And basically the core ideas were that standard nitrogen fertilizer recommendations do not account for management-induced variation in mineralizable N, but I believe that they should. Soil test biological activity is strongly associated with mineralizable soil nitrogen. So ideally, we would want to measure mineralizable soil nitrogen. That would be the key thing. It would be direct. We'd be measuring nitrogen, but because the nitrogen mineralization occurs because of the activity of the organisms, we can simply measure an easier parameter, such as this 3-day measurement. And that gives us an idea that mineralizable soil nitrogen will occur. So nitrogen fertilizer recommendations can be adjusted for field-specific level of soil test biological activity. And that's shown here in this diagram, where if we had low biological activity, we need to follow the recommendations basically for what I would say, just to use, a simple term, is relatively dead soil that doesn't have much biological activity. It's just not living to its level because it doesn't have the resources there. But when we give soil resources, organic resources, then we have higher biological activity, and it's mineralizing this nitrogen on a continual basis, leading to lower level of need for this nitrogen fertilizer. And so we could be making adjustments to that nitrogen fertilizer application. Well, I wanted to not only focus only on the yield response to nitrogen, because although that is very important, and many farmers tell me that's really good information, they want to know about it. But biological activity actually has something to relate to other functions of soil, such as water stable mean weight diameter, and this would be just simply soil aggregation. In this case here, there's somewhat of a weak relationship between aggregation and biological activity with conventional till cropland. But if we add in no-till cropland into some of these responses, then we can see, well, okay, there's a stronger association just in the no-till cropland, but it's elevating the aggregation per unit of biological activity. We add in some grasslands, and we can see there's a good deal of scatter, because it's not an absolute, because aggregation is not only determined by biological activity, but also by mineralogical features as well. And so there is an association. And then we add in woodland, and we see that there's a lot of scatter here, but in general, we can see that there's a common relationship that's occurring, and it seems to plateau at some point, and that would be good in some ways, because we don't want to have excess biological activity. We don't want to have purely organic soils that are fresh organic matter. We want to have them stabilize over time, and that leads to what I would consider a very stable aggregation that occurs. And so biological activity can indicate that. My final part of the program is to relate to you how I'm taking this information on biological activity and storage of carbon in soil to this depth association. So the title of the presentation is that the activity starts at the surface, right? And so the surface organic matter is really, really important. Now I'm going to focus on the top 12 inches of soil, or the top 30 centimeters, and relate to you some observations that I've made over the last 5 to 10 years. And I put this into what I'm calling root zone enrichment as a way of doing this. So the vast majority of upland soils have this nonlinear depth distribution of soil organic matter. There can be some variations, but in general, it's really finding this nonlinear association. So these are some data that I pulled from some old literature when I was down in Georgia. I had these data, and then I kept looking at them year after year, figuring out what I could be doing with these. So these are not data that I collected. These are from a soil survey collected all across the state of Georgia. And you see that in the red, in all cases, all land uses here, there's this concentration near the surface, and it declines with depth. And what was obvious to me was that they're all converging upon some level of organic carbon with depth. And that depth is anywhere from, say, 20 to 30 to 40 or greater depth, where they're converging. Land use doesn't seem to make any difference below that depth. So focusing on the top 30 centimeters, we can say that that's where any change that did occur, it's going to occur near the top, in the top 30 centimeters. And there could be some change that occurs due to management with increasing depth over centuries. But in our general lifetime, in a generational lifetime, we're not going to see, it's my hypothesis that I don't think we're going to see any major change in organic carbon with depth below 30 centimeters due to our management activities in a generation time. Maybe in centuries, but I'm not sure that we're planning for next year sometimes, and thinking about centuries, it would be a harder sell for some of these ideas. So below 30 centimeters, there's little solid evidence from this and other studies for change in soil organic carbon due to management. The management is the important part. There's always carbon below 30 centimeters. We can quantify that, but that's due to pedogenic process. I'm going to give you some examples from some real observations that I made from my own. And so this is the soil organic carbon with depth under a situation. It doesn't really matter what situation it is, but I wanted to describe the process of how we are starting to quantify this effect. So this is the mathematical description. We have soil organic carbon, whatever that concentration is. It's going to be a function of a coefficient here, a coefficient B. It's nonlinear, exponential function with a rate constant and soil depth. And the A function, the quantity there, is really the carbon that's deep in the profile that isn't really going to change. Theoretically, if we go deeper, of course, it likely will continue to decrease in concentration. But for all practical purposes, anywhere from 30 to 100 centimeters, we should be able to come up with a number that is essentially not changing. The B function is where we really want to focus our effort to quantify what is the effect of land use, or in more specific terms, management, how the farmer is, or any land manager is concentrating their effort on changing what's in the upper 30 centimeters. So we can see, then start to quantify this, that the concentration of organic carbon at 30 centimeters can be determined from this nonlinear regression. And we then determine a polygon there, which is the baseline. That's what I would call a petagenically controlled carbon accumulation. And that needs to be subtracted from what I'm calling the root zone enrichment. And that change that occurs between the total and that baseline condition is the root zone enrichment. So we want to quantify that and give it a value. Now, I would consider this an index, but it can also be quantified into an absolute. And I think we need a whole lot more data to actually start using it if we want to eventually use it with the term sequestration. But basically, it's an index right now, and that is partly the reason that I continue to call it root zone enrichment. Now, we have another curve here, and these were taken from a data set where I had a statistical distribution of the 5 to 95%, and they're all taken from North Carolina in this case. But we have two different baseline conditions, because at 30 centimeter, we have that regression line converging in different places. And so we have two different root zone enrichments that can be calculated from these two different examples. And you can see here that the baseline is very different, anywhere from about 16 tons per hectare versus 47 tons per hectare. So that baseline is very important for us to determine. But the root zone enrichment, although it is different in these two cases, it's not nearly as different as we would have expected it to be, because we've subtracted off a vastly different baseline condition. And that baseline condition can be taken off of neighboring sites, or it can be taken off of two different farms in two different counties. So the root zone enrichment is an important process because we want to avoid this condition of this photo here, right? We want to change our land use so that it's not destroying soil, but conserving soil. Degradation of soil from tillage-induced or tillage-intensive agricultural management is widespread and still widespread, and we need to control this. Excessive tillage and lack of sufficient organic resource inputs contribute to low soil organic matter and severe erosion, leading to poor soil health and low productivity. And reinvigoration of soil is possible with conservation management. Conservation management is what we need to have, and we need to convince people that that's what we need to do. There, I have a hard sell in the coastal plain, because we don't have necessarily intensive erosion that occurs because it's a very flat landscape, but it still does occur. And it's also degrading some of the resource capabilities of the soil to function. And so we still have a lot to do in the conservation world. So some other additional background is that organic resources are primarily deposited near the surface. So that litter from, say, whether it's a woodland or grassland or a conservation till cropland, it's being deposited near the surface, and then it's changing the surface conditions. And soil below 30 centimeters is often more consistent in temperature and moisture over time, thus favoring decomposition. And of course, I'm taking this from my own example in the southeastern US. We don't really have freezing soil temperatures other than the first couple centimeters. And we basically have conditions deeper in the profile that are favoring decomposition. And that's primarily why we don't have much organic matter deep in the soil profile. And so there are these conditions that we need to be aware of. Deep profile changes, that is below 30 centimeters in soil organic matter, will be magnitudes slower and smaller than at the surface. And so we can expect that perhaps over decades, centuries, we could possibly change organic matter deeper in the profile, but it depends if we're going to then apply carbon below there, because if we're not applying it, even deep rooted crops aren't applying much carbon deep in the profile. Plus, then they're also, that carbon is being decomposed, and carbon, soil organic carbon and total nitrogen are a function of both input and the respiration that decomposes it. All right, so there are some key considerations that root zone enrichment of soil organic carbon and nitrogen fractions focus on separation of contemporary management influences from those of pathogenic origin as a concept. And it's a concept right now that is getting populated with data. A mathematical function and not necessarily biophysical in nature. This is why it's an index, because I'm trying to describe natural processes with a mathematical function, and in general, it seems to work, especially under undisturbed conditions. Whenever we disturb the soil with inversion of soil and tillage, we can create abnormalities in that depth distribution. But if we stabilize it a little bit over a couple of years after this conversion of say moldboard plowing, we can actually still fit these functions to it. And there's advantages of relatively low cost and practical applications to evaluate effects of land use change. And that's the key here, is that we're trying to understand what are the implications of land use and management over long term. So there are some assumptions too, and you may or may not agree with some of the assumptions. And if not, then of course we want to have evidence. We want to have a good discourse about this. Pedogenesis will have a large overriding influence on base level of soil organic matter. The soil formation factors that occur across the landscape all around the world are important, and this is one way where we can account for them rather than, of course, we still need to understand why they occurred, but this is a simple index where we can account for them. If historical management were destructive, and I have examples in the southeastern US, in the Piedmont, we have enormous amounts of historical erosion, and this is a true fact here, then residual surface organic matter would be low, and the new soil surface might be more like that of the original subsoil. We can see evidence of that where we have extremely low organic matter at the surface because it's been eroded away. Bulk density of soil throughout the profile is considered inversely related to soil organic carbon concentration. This is a factor of kind of a fact already that from data sets all around the world, we see this inverse relationship between soil organic carbon and bulk density. And this is an important part because we need to have the bulk density estimate in the calculation of the root zone enrichment. Soil organic carbon and nitrogen concentrations at selected depth intervals can be reasonably characterized, the actual depth distributions. Okay, so fitting that function is important, and so we can be able to do that with these depth distributions, and it seems to work in general. I'm going to give you a few examples to end the presentation here. And the idea here is really taken from soil organic carbon, but this can be applied to soil test biological activity also. It could be applied possibly to other organic fractions, and not even fractions as such, but organic indicators. It could possibly be used with enzymatic activity or other metrics. I'm not sure it can be really used with microbial community structure, but it might be because of course, resources are concentrated in their surface. So it's the concentration of resources. This example here, if we measured only the total stock under conventional tillage and no tillage, and this happens to be a Tidewater research station near the coast in North Carolina, and we have highly variable soils because we have both organic and mineral soils in the same landscape. In this case here, if we measured the total stock, from 4 different fields on this research station under conventional tillage, and then 4 fields under no tillage, we would say that conventional tillage led to greater stock of organic carbon than no tillage, which doesn't seem to make much sense because that's not the way conservation typically works. Although if we, the literature is full of this dichotomy, where some people show that there are no differences or even opposite effect, and then we get this mixture. But there may be a reason for that. And in this case here, the baseline stock, if I calculated that with this root zone enrichment approach, there's far greater baseline stock under the conventional tillage than no tillage. And it leads to the conclusion, in my conclusion, would be that no tillage is still storing more carbon due to the management rather than pedogenesis. The management has created more carbon stored than with conventional tillage. So that's an example of how we could use that information. We go to another research station and we see that there's really no difference between conventional and no tillage, and the baseline stock is no different. So it ends up that the root zone enrichment is no different, and that can be a logical outcome. It's not a problem. We go to the upper coastal plain research station. It's in a similar location as the Cherry Research Farm, but it's not. It's a different location, and we see that no tillage has far greater carbon stock than conventional tillage, but it's partly due to the baseline condition. But if we make the root zone enrichment calculation, we also still get this conservation effect where no tillage leads to the conclusion that we would have more carbon stored due to no tillage than to conventional tillage, as a function of management rather than just the pedogenic feature. Go to another site, we have no difference in the total stock, no difference in the baseline, and no difference in the root zone enrichment. Sometimes we get too much variability and we can't overcome that variability. In the Piedmont Research Station, no difference in the total stock, no difference in the baseline stock, and no difference in the root zone enrichment. But you're seeing a trend in a couple of these cases where there's a trend for no tillage to be greater than conventional tillage. And in the going up to the mountains, we see no difference, but in the root zone enrichment, there actually is a difference. And again, that's due to the baseline stock. And if we put them all together, if we had this all together, we would say that there's no difference in the total stock of carbon between no tillage and conventional tillage, when in fact there were some differences in the baseline stock, leading to what I think is the truer conclusion is that no tillage did store carbon greater than conventional tillage due to the management if we take out the pedogenic factor. And we can do this with the biological activity. This is just the example I'm going to give to you as a biological activity. We had 3 private farms in this case. They all 3 farms had a cropland, grassland, and woodland fields, multiple fields in each. And you see the number of fields here. Of course, we had more in the grassland. It was partly because I was more interested in the grasslands at this case. But the total stock was no different among all of them. But we did see that the root zone enrichment there was a difference between grassland and cropland. And there was a trend that there was greater in grassland than woodland, but it did not come out statistically different. So again, this biological activity can be used with this technique, and it can show some real differences. So I wanted to just wrap it up, and I've given you kind of 3 different aspects of the presentation, just the simple determination of biological activity and how we can use that. And then using it to predict nitrogen mineralization and its ability to shift the amount of nitrogen that might be required for typical grain crops. And then finally, this root zone enrichment calculation that can be used with biological activity, but is more practical for, say, organic matter, total organic matter, soil organic carbon, total nitrogen. And total nitrogen is of course very interesting for farmers because it is the warehouse for getting their fertility. All right, so soil test biological activity is sensitive to edaphic, environmental, and management factors. And so we can be confused sometimes to which factor is it really attributable completely. And so we need studies to kind of tease that out. And the responses of soil test biological activity to management are reflective of the guiding principles of soil health. So using the 4 soil health principles that NRCS has outlined, these are really important because they do lead to changes in biological activity. The results of soil test biological activity can be used to modify nutrient management and particular nitrogen, but I believe there's opportunity to actually start looking at phosphorus, because phosphorus, organic cycling of phosphorus is significant, but we've put a lot of emphasis on the mineralogical portion of that, to the more physical portion of that phosphorus, or biophysical with the chemistry as well. But there's potential to change phosphorus recommendations too, I believe. Strong depth distribution effects require careful collection and interpretation of results. I think that's just a simple thing that I want to leave you with is that we do need to be careful, because the measures are sensitive, and so we don't want to just flood the literature with poor values. So with that, I leave you with my email if you wanted to contact me afterwards, but otherwise I'm going to unshare my screen and leave you with that there.
Wonderful.
Thanks
you,
for the time.
Alan.
Yeah.
I appreciate that. It looks like we have some questions coming in to the chat, and I encourage everyone, if you have additional questions for Alan, to put those in. Let's see. We have a question here that two folks had from Marcel. So how does this test relate to the Hanley test results?
To the Haney test? Yeah, it relates pretty well. The Haney test is its incubation time is 1 day, 24 hours, and there are some differences, let's say, in the laboratory approach, and that can cause some modification of the association, but essentially we haven't really done a good job of characterizing the association across a very strong diversity of soils. We've done it in a few studies where there is some association, and if one has used the Haney test over time, it's probably good to keep using it, because anytime you change methodology, you're gonna change the results. But unfortunately, or fortunately or unfortunately, the labs are also adapting to new information, in that the Haney test is really developed on the idea that you add the water from the bottom, so as a capillary action, and that can actually saturate soils, particularly sandy loam soils, which we have most of in North Carolina. We have a diversity, but we have a lot of sandy loams, and we actually it leads to a little bit depressed carbon mineralization, and so the numbers aren't necessarily what we might expect. But overall, it's based on the same principle.
Okay, interesting. Thanks. Ashley Wagner has a question. Some soil health tests for respiration only measure after 24 hours. Is there a reason you suggest 3 days instead of 1? Yeah,
as I was explaining, the Haney test is a 24-day test, and of course, Rick is no longer as he's doing his own things now. Outside of, he used to work for USDA ARS in Temple, Texas, and we went to school together, so we both were working on this. We realized that he realized that he thought he could make more progress by using 1 day, and so just because it's quicker. But I don't think quicker is, in this case, is necessarily better. I think we get a more robust number. Of course, the way he would do it is with an infrared gas analyzer to detect the CO2, and that's a logical and a straightforward way to do it. It gives us good information, but the longer the incubation, the better the number will be. We just get less variation. I mean, that's been shown before.
Okay. Claudia has a question. This may not be within the boundaries of this presentation, but would we see a greater benefit to soil with deeper rooted plants?
Deep-rooted plants are important. Don't get me wrong, but even when we have a native warm season grasses, which are notorious or well known for their deep rooting capability, they do provide carbon input deeper in the soil profile, but the amount of carbon that they provide is relatively limited. And this is why I say that, things can change over decades to centuries, but it's not something that we're going to observe immediately. Having deep-rooted plants the plants get more water from deeper in the profile. That's absolutely a good thing. Does it really affect the health of the soil over, say, a 5 or 10 year period? It may be questionable. But I think over many decades and over centuries, for sure, it will lead to positive changes. That I'm pretty sure of. But we won't be able to measure it unless we had some of those fields so that we could go back, you know, like a sequence where a chrono sequence where we could actually do that. And that could be possible. I just think that deep-rooted plants are important. Let me just state that clearly. They are important for getting the soil to function properly, because that water conduit is really important, and getting plants to produce more so that they can actually deposit more in the soil, and that cycle continues, is really important. So that will occur, but I don't know that we're going to change in a reasonable amount of period the soil health condition.
Interesting. Thanks for that. Ben is curious if you could comment on the value of the on-farm Solvita test.
Yeah, Solvita. I know a little bit of history. Rick Haney helped the Solvita develop the test, and so that it's a color paddle, and so the in-field test, which you can do the test in the field, where you take a sample and just use the moisture and temperature conditions in the field. Those moisture and temperature conditions are extremely important in getting a number. And when they vary, if the soil is a little bit drier, okay, you're not going to have as much activity. If the soil is a little bit too wet, you might have less activity also, because it's hard for the CO2 to get out of the soil. So all of that, those things are important. If it's cold, of course, the soils don't respire as much. It can be useful if you can continually use it, knowing the conditions, but those conditions vary, and so it's hard to use the numbers in a really logical manner just by themselves.
Yeah, great. That makes sense. Bob is curious, one of your slides showed pastured cattle. How effective are dung beetles in the redistribution of organic carbon compared to plant roots growing in the same field? Any idea? That seems like a specific question.
Yeah, it's a great question. I haven't made dung beetle measurements myself, but knowing the different species of dung beetles, of course, is important, because we have some that actually burrow deeper and some that are just essentially at the surface. So there will be a diversity of beetles, and so that will matter. But certainly the fact that they're making the pat disappear quicker means that carbon is then getting incorporated into the soil and could have a more immediate effect. And so I think there's a possibility that it could be really effective. The roots are always going to be following these channels too, because it's looser soil. There are nutrients that are then being mineralized. And there are plant growth promoting compounds that are being produced. So that, yes, dung beetles should have a positive effect.
All right, great. Well, if there are any other questions, please go ahead and put those in the chat. You can also, another option for folks is to unmute. You can unmute yourself and ask a question that way. You're getting some thanks, some kudos on this great presentation. So we really appreciate you taking the time here today. Yeah, some claps. Wonderful. I'm just trying to see if there's anyone other questions coming through. It does look like that's the rest of the questions for today. We have a little bit tight on time, so I think we're going to go ahead and end now, as opposed to taking additional time for breakout rooms. Hopefully there'll be time for breakout rooms at a future digital cafe. I did want to share my screen here and just note to folks that we do have a talk coming up next month. August 21st is going to be the talk, and we're going to be talking about helping growers start with certified crop advisor Lee Bryce. So excited for that one. That'll be a different perspective from folks, from him, And he's a well-known speaker who's generously donated his time to join us. So be sure to join us for that one. You can find the registration again on our website at soilhealthnexus.org. And see, lots of thanks. I don't know, any other final call for questions for Alan? See, like maybe someone's trying to unmute here. No. All right. Well, thank you so much, Alan. I really appreciate your time here today. And thanks everyone for joining us.
Thank you all.